delighted to welcome Dr. Raphael Valdivia. He is Associate Professor of Molecular Genetics and Microbiology at Duke. As the Vice Dean for Basic Science, he serves as a liaison between the Dean's Office and the basic science community, and also provides oversight of the School of Medicine core facilities, research lab space utilization, and the biomedical graduate programs in the School of Medicine. And today is going to be talking to us about strengthening the ties between the basic science and clinical research communities. So please join me in welcoming Dr. Valdivia.
Thank you, Emily. I think this is on. Can you guys hear me? Okay. Do you guys usually sit all the way back? Is that kind of how it works? That's all right. Just a couple of words to clarify my position. So I'm certainly the Vice Dean for Basic Sciences, but that is my night job. My day job is as a researcher. I'm a faculty member in molecular genetics and microbiology. I have a lab in the Jones building, which is a little bit down the road. And 60% of my effort is devoted to research activities, and I'm a basic scientist. And my role in the Vice Dean office is kind of to expand the role that the basic sciences play at the school at large and how it interfaces with basic science programs in clinical departments, and then eventually, as the title alludes to, as to how to strengthen the connections between those basic science programs and the clinical research and the translational world. So I figured I would start first by giving you an introduction about the School of Medicine. You may know this, but there's 2,200 faculty at the school and another 5,000 staff members. The organization of the school is such that there are 7 basic science departments, 15 clinical departments, and 18 centers and institutes, including the DCRI. And they're all spread around campus in over 93 buildings, also in downtown. And I'll talk a little bit about the efforts that are expanding our footprint in the downtown area, both for research space and translational space. The annual budget of the school is a little bit over $1 billion with around $600 million in sponsored research. So this is a very large machine. We generate a lot of research output, mostly in the clinical area. Clinical research is really the largest part of our portfolio, and probably around 4/5. I should also mention that among our faculty, we have a substantial number who are in the non-tenure track, and most tend to be in the clinical departments. The basic science departments tend to have more classical tenure track type lines. So in a way, the culture that has been created in both basic science departments is closer to what you find in a normal non-medical school university. Now, having said that, the basic science departments, even though they have a core of faculty who are really embedded in that discovery science world, there's very strong basic science programs within clinical departments. And in fact, there's more faculty participating in basic science research in clinical departments that are in the basic science departments themselves. Okay, so my office, what is it that I do? And my main role is really to be the advocate for basic sciences at the school. As Emily alluded to, I oversee the biomedical graduate education program. I provide strategic support for share resources and core services. These are the core facilities. I try to, and as much as I can, to bridge the basic science research efforts across all departments, and not only in the School of Medicine between clinical and basic science, but really across the school. So that includes arts and sciences, the School of Engineering. More and more there's a robust growth in biomedical engineering within the Pratt School. Many of them very, very much interested in clinical applications and also in big data science, which we're very rich at producing. And so with Sally Kornbluth as the new provost, there's been this renewed effort and trying to bridge the two together. So there's a lot of opportunities that are going to be coming up. And in this position, it has given you a bird's eye view of all the research activities happening across campus. But interestingly enough, not as much into the clinical arena. So in as much as I'm coming here to tell you a little bit about basic sciences, I'm also in the learning curve for trying to understand what is happening in the clinical research world. I also get to hand out cash, so this is kind of a fun part of the job. I get to administer awards and pilot programs and funding opportunities that come up from both philanthropic sources, but also from federal sources and foundations. And so that is actually one of the fun parts of the job, because when you send somebody a notice of award, it's amazing the great things they will say about you. Not as much when you tell them that you don't have any money for them. And then I also have to do some tasks that I just keep the trains on time and running. That's oversight of the animal programs, research space allocation for basic science programs. So when people need a lab space, they usually come to me and they say, give me a lab, which is not as easy as it may sound. And they also oversee conflict of interest and research integrity office. So this is kind of part of a larger portfolio. I wouldn't say that these are the fun parts of the job, but on the other hand, it got me to understand what goes into operating a machine as large as Duke. And then there's a lot of components that need to work together, and especially in the area of research compliance and research integrity, to understand some of the challenges that investigators are facing across the board. So what does it mean to be an advocate for basic sciences? And this is really the reason I took this position, is I very much appreciated what other people had done for my career, for my research program, and that for me to be in a position to give back and to help their programs is really a unique opportunity, and really something that I very much enjoy doing. My main goal is really to ensure that discovery science remains a priority at the School of Medicine. And so, this concept, and I will talk a little bit more about this, about what basic science really means, it means different things to different people. But the way that we're trying to distill it in a way that everybody understands, is really about discovery, learning new things, and that should be really a priority. And so all the faculty that are involved in this process, the basic science faculty and the research programs, my plan is to support them. And that means removing barriers to innovation or anything that gets in the way in terms of compliance issues that are really self-imposed. I'm there to highlight their accomplishments and promote them. And so that is really where I get to work the closest with the chairs and division chiefs to achieve that. And also, as part of this innovation aspect, it is my role to support cross-disciplinary efforts and grassroots efforts that strengthen the quality of our scholarship. And so what that means, and whenever you get a group of investigators deciding that, hey, we have an idea, and I think if we get together, we can do this, we just need a little bit of support, we try to make that grow. Or whenever I see from this position that somebody's working in an area and they're completely unaware of somebody else in a clinical department or in the School of Engineering that is doing something similar, I can be the conduit that can say, hey, you guys need to talk. And it's remarkable how often that has led to very interesting new ventures popping up. Another role that I have as an advocate is just beyond what I do at Duke. For the research laboratories, I think, and this is a trend that certainly we've been seeing in the last couple of years, where more and more scientists are getting involved in trying to inform legislators and the public in general as to the value of what we do. And so being strong advocates for research funding is, I think, an important task. And I view all of us as scientists as also that we are citizens. So we should really be involved in the direction of our research enterprise. And so we're happy to say that through a collective effort, not only just by schools like Duke, but also through professional societies, I think that we've kind of turned this bipartisan support for research funding into an actionable material, such that we've been getting sustained growth in the last couple of years, despite previous dips in funding. And so that has also involved going to Washington, which is kind of, for me, been a very different beast. But I've kind of got to learn a lot from doing this, and the importance of having this face-to-face connection with decision makers. And as much as we would like to think that people are data-driven, turns out that people are very emotion-driven. So if you can get in front of somebody and tell them a story, a compelling story, that can make a lot more of an impact. And so for the last several years, I've been taking faculty members from Duke with me to Washington to to talk to the North Carolina legislators about the importance of not only the research that we do, but also our impact to the overall North Carolina economy. And I think those are very well received. And so, those opportunities are going to keep on coming. And when you see the announcement, please make sure that you contact Catherine Liao, who's usually my contact person for arranging these kind of trips. And so we were very lucky to have Vice President Biden come and visit last year as part of the Cancer Moonshot, visit the Duke campus. Many of you are probably aware that he was in town because the whole place was shut down and there were snipers on the rooftops. But this is great. I mean, this is a great way for us to showcase the quality of the research going on here. He probably came here mostly because of a lot of the work that was done in the context of brain tumor treatments through immunotherapies, work done over here through the neurosurgery department. And he also got to meet here with our latest Nobel laureate. And so this is just Paul Moderich was just a fantastic guest. For those of you who know Paul, I mean, he's a very he's not a very outgoing person, but he became very animated and really trying to sell this idea of discovery science to vice president, and he was definitely a convert. And so we're hoping to keep on building those relationships. And that is part of the job that is different. It's something that I was not trained to do, but it's something that I can be learning along the way, and I appreciate the value of it. Okay, so one of the things I wanted to accomplish today is to introduce this concept of basic science, and I alluded to the fact that this means different things to different people. Me, as a basic scientist, this is kind of how I view basic science. It is really mostly a focus on discovery. We are explorers of the natural world, and we just want to try to figure out how things work. We're driven less by this having eureka moments, which essentially is just an acceptance of knowing that something that you thought was true is true, which is, okay, satisfying in many aspects, but really the drivers, the excitement that gets created when you find something that's completely new and unexpected, right? So when you go the moment, those are the ones that really drive innovation. And so, for many of us, the goal is really knowledge for the sake of knowledge. So this is kind of like the baseline, but in reality, most scientists, basic scientists, are really across a spectrum, right? On the one end, you have the people who say, don't ask me how my research is translatable, because they're just sick and tired of being asked that. They just want to know how it works. This is the kind of person who grew up breaking down the radio because they want to figure out what was inside of it, not necessarily with any intent of putting it back together. In the middle, there's more of the people who are doing their work and that they know that there's some disease that could potentially be helped by the research, but it's not really their main driver, right? That's not why they do things. Other people are very interested in the basic aspect of a pathway, let's say, that they're studying, and then they know that there's some diseases that are associated with them, and they start thinking, if I learn how to stop this, maybe there's a therapeutic value to it. But if their interests change to a different pathway that is not associated with disease, they'll study that pathway. They won't stay on the pathway just because there is a human disease associated with that. And then there's the other group that I would say that are more like driven by the natural questions that emerge from looking at human populations, right? There's a disease out there in a particular set of patients, and they view this as a question, right? The same way that you want to understand how a protein folds, they want to understand why is it that a particular group of people get sick and to find out what went wrong with them. And that is the kind of questions that they want to ask. I put the gradient from white to black, not necessarily to mean that there's a darker side to one versus the other, but just to say that it's really a continuum. And I would say that for most faculty who are in a medical school setting, they're somewhere in the middle. And there's a couple of thoughts that I also wanted to kind of throw out there as to maybe some misconceptions that we have about basic scientists and what drives them. And I think sometimes people who are very much in the clinical and translational setting, they tend to think about, well, why are you doing what you're doing? What is the application? This is the kind of conversation sometimes we also have our Thanksgiving dinner, all right? It's like, your family members asking you, and you're going to cure what? And what you're doing is going to lead to, and in the reality, really science should not be just viewed through this prism of is this translatable, what is the application? Because in fact, we can't often predict what our findings, what translational application they will have. I mean, so one of the most salient examples right now is CRISPR gene editing technology, right? So this arose from people studying bacteria in brackish waters and how they prevented phages from attacking them. Those people studying that were not really driven by how can I edit genes in mammalian genomes or do gene therapy. Sometimes the findings that we have in the laboratory are best translated by other people who can take advantage of that knowledge. So in fact, basic scientists are not always the best at finding applications for the discoveries. But also for us basic scientists to understand that the questions that come up in the clinical realm are not dirty because they came from a translatable or an area that is viewed as applied science. In fact, there's a lot of fascinating basic science questions that are coming out of the clinics all the time. All right, so with that setting as to what I mean by basic science and the basic science culture, and that sometimes clinicians can find it hard to understand. Why is it that your whole lab is studying X when Y is a really interesting thing because it will allow me to treat patients with a certain disorder? No, that is not what the drivers are. So in as much as one can find areas of common ground, I think the more productive people can be. It also has to do with training. And so for this, I'm just going to segue very quickly to one of the areas that I oversee. This is the Office for Biomedical Graduate Education. And so this is the office that oversees all the PhD programs in the School of Medicine. We have many of them. We have over 600 PhD students across the school. And they range from all kinds of, from pharmacology and cancer biology to biostatistics and bioinformatics. And the training that we give the students is really focused on their labs and the research that they do in the labs. And as such, in a way, we are perpetuating some of the habits that we had as basic scientists, where we do not know the clinical world, we do not know the translational world, so how can we train them really to be more effective and bridge those areas? So there is a program, the Duke Scholars in Molecular Medicine, which is currently run by Nancy McIver, and before that by Pat Seed, who started this. And this is an interesting program that allows for PhD students to shadow a clinician for a year. And there's different areas of focus in infectious diseases, cardiovascular medicine, neurosciences, nutrition and metabolism, to name a few. And the idea here is that you really get immersed in the kind of problems that come up in the clinics. And this has just been just mind-opening for people. Sam, did you go through that program? No. But we had a lot of our PhD students who were very much set on basic science careers who suddenly decided, you know what, actually there's other areas that I find of interest and whether it is in not only in clinical research, but also in science communication and more in the translational world when it comes to devices, really a better bridge between the basic sciences and the more translational sciences. And so there's, this crossroads between clinic and the lab, which they were not even able to see before, suddenly now they're able to have that choice. So this is one area where I think working together with our clinical faculty, we've been able to expand the set of options available and enhance the training of our PhD students. And so a lot of these things we're doing in conjunction in partnership with the Translational Sciences Institute, and one of the big parts of what they do is discovery science and how to plug it into best with translational applications in clinical world. And one of the things they support is this accelerator program. And so this is encouraging basic scientists to maybe grab some of the projects that they have on the side that may have a translational application and support it through pilot granting mechanisms. I mean, so this is in this particular press release here. This is Brian Cullen, who's in my department, molecular genetics and microbiology. He's become very good at doing gene editing approaches, and so they got funding to see whether they could cure some chronic viral diseases by applying gene editing tools. So another way that we try to link basic sciences and translational sciences is through shared resources and core facilities. There's over 70 of them across campus. They're not owned by the School of Medicine, but they're owned by their entities, because they are the ones who know best what kind of shared resources they need, right? So I'm not going to tell, it makes a lot more sense for a crystallography core to be within a biochemistry environment or an institute that does a lot of structural work than it is for me to try to oversee it. And some of them are really at the interface between basic sciences and clinical sciences. We have a new core facility devoted to preclinical studies, and so this is the kind of people who would help basic scientists develop the animal models that they will need to test various interventions, for example. And really, overall, the goal of this core facilities is to make these new and cutting-edge technologies available to all investigators. And so that not only means basic scientists delving into clinical sciences, but also clinicians beginning to do work in basic sciences. So for example, we have a microbiome core facility, so such that if clinicians are beginning to look at stool samples, maybe associated with some particular ailment, then they can get the support to try to derive data from those samples in a meaningful way. So they can really become conduits for translational applications. The cores are also help with the proper experimental design and data integration, so that's also very important because most people are not really trained to do all aspects of research, right? I mean, no, we are not all genomicists, we're all not microbiologists, and so more often than not, you have to rely on the expertise, and which you can do through collaborations, but having a core facility that is staffed with the people who know what they're doing, I think is very important. The other place where the cores play a pivotal role is an integrator of new ideas. And so we've seen this already for a lot of research projects where, one investigator will go to a core facility and wants to do things in a certain way. They will come in with a manuscript on a paper and say, I want to do this. And then the core facility director, who are all PhD level scientists, can look at these experiments and say, well, actually there's a better way of doing it. And so in pharmacology is doing it. So suddenly, how you approach your research question changes based on new information and new expertise that becomes available to you. So this is an area where the chancellor has come in and helped invest pretty heavily on upgrading our shared facilities, and that includes a $31 million commitment over the next 5 years. And thanks to support from the chancellor and from the dean, we have a notobiotic core facility now, so you can raise animals under germ-free conditions and do the kind of causal experiments that you want to do as to whether a particular set of microbes or a particular set of microbial communities associated with any one disease. We have animal pathology services to supplement this and to many of the other. This is the first time we've had a board-certified animal pathologist on staff. And so now you can look at your animal and understand what exactly is going wrong with them. We have a viral vector core that just started through neurobiology, and this is another way of, in an experimental setting, manipulating larger animals. We had major upgrades in our proteomics, genomics, and sequencing cores, and I'll come to that in a second. This is really what is driving a lot of science these days, doing very large omics types of studies, and certainly in the clinical arena, that is probably this is the future. Clinical genomics is where you're going to be deriving a lot of your data for future experiments. We have major upgrades to a light microscopy core facility, and we're about to launch our first cryo-electron microscopy facility, which is a triangle-wide effort. So just a couple of words about what are some of our flagship cores. These are the sequence and genomic technologies cores. So this is the people who do all the deep sequencing, next generation sequencing, single molecule sequencing technologies. We have a proteomics and metabolomics core facilities. These are very large operations. They are really packed tight on Duke campus. And so we decided to invest in them heavily, expand them, allow them to grow. And so they're going to be moving downtown to the Chesterfield building. For those of you who have been on Brightleaf Square, you've probably seen the Chesterfield building. It's been wrapped in saran wrap for a little while it took all the asbestos out, but it's been revamped. This is actually a picture that we just took a week ago. It's going to be a beautiful space. 2 floors are going to be Duke. One floor is going to be an incubator company. And so this is a place where you're also going to be able to have incubators, ventures being right next door to do research programs. And so the core facilities are going to be going there. They're going to be able to partner with the companies that manufacture the equipment and really, I think, turning into the technology innovators that we would like them to become. So this last year, the Chancellor released a health system strategic plan. The School of Medicine released their own strategic plan, and this has become the strategic priority as it relates to basic sciences. And so there's 6 main points. The first one is, and the foremost, is to maintain this world-leading faculty for biomedical research at Duke, and to recognize these individuals, develop them, and recruit and retain outstanding talent. I mean, after all, really what makes Duke Duke and where it makes it the great place is the people that are in there. And so this is the greatest resource we have and the one that we had to nurture. And so it is critical for it to be at the top of the strategic plan. They need places to work, so we need to strengthen the research infrastructure and share resources. I kind of mentioned a little bit about that. As I mentioned, research is moving more and more into the quantitative realm, so we need to give them the expertise to do that. Most basic scientists and most clinical sciences don't come necessarily with a very strong training and to allow them to make the best use of the data that they have available. We want to keep on nurturing this culture of discovery and innovation and high risk. We want to be at the leading edge. High risk leads to high pace. And then overall, just generate a vibrant culture that makes it easy to conduct research and integrate research, education, and practice. Because it is not really about only us performing the research, but also training the next generation of people. So in terms of infrastructure, we have the first new research building in over 6 years, and this is Medical Science Research Building number 3. This is on Research Drive. You've probably seen it go up. This is just from 3 weeks ago. This is the last beam that goes somewhere over here. The building is being planned. 2 floors already been occupied with programs, and then the rest is being left available for a couple of research initiatives that I will touch on in a second. So we also launched this molecular microscopy consortium. This is, again, a huge investment, almost $10 million, that are going to go into really bringing this really cutting-edge technology into the Triangle. Mario Borgnia is going to be the director of this center, and it really represents a true partnership between the NIEHS, Duke, and UNC to allow for the molecular analysis of protein structure in a way that is not done before. I mean, crystallography, some people argue, is kind of phasing out, and cryo-electron microscopy is going to be what it replaces it. I'm not a crystallographer, so I'm pretty sure some people would object to my portrayal of the future, but this is kind of where I'm understanding how things are going. So a lot of expensive machines, a lot of infrastructure, IT infrastructure to manage the massive amounts of data that are going to be coming out of this. And what they really essentially do is grab complex macromolecular structures, in this case an example of viruses, either through cryo-electron tomography or just single particle analysis, and through just sheer crunching of data is reconstruct images to atomic resolution. And so when you start thinking about mapping antibody binding sites on a virus, or where a drug fits into a particular conformation of a protein, this is really where the future is going to be going. Because you're not restricted to a snapshot of a structure, but you can actually follow it in almost in a dynamic process. So this is exciting. It is being launched starting in April. And so this is really the next wave in technology in this area, and we're going to be at the forefront. So as part of the strategic initiative, the Chancellor put out a challenge out there for various faculty members to come together in particular areas of focus that he wanted to drive as a starting point for the next generation of innovation. And there's 4 areas that he concentrated on: cancer, cardiovascular disease, transplant and immunology, and neurosciences. And so what he did is he charged people to come up with these white pages of next great ideas where there's going to be a bolus of resources put together to let them to launch. And so I'm just going to very briefly talk about 3 of them. So these are kind of things that are up and coming. One of them is in the area of transplant and immunology. And this is a group of investigators, both from clinical to basic science, where they kind of want to redefine what homeostasis is in terms of immune health, and use that as a parameter to look at the spectrum of what it means, what is tolerance and what is immunoreactivity, to try to address issues of immunity to infectious diseases, and also immunity self-recognition in terms of autoimmune disorders, but also in terms of how patients will respond to transplants. Another one, big focus is in the area of cardiovascular disease. This is a little bit slightly different because it's more focused on populations. And this concept that we are really on a spectrum between health and disease, and that there's a lot of different factors that can determine whether we go in one direction or the other, certainly our genetic composition, but also lifestyle, environment, injuries. And so where we are in any one spot, to understand how that occurs. What is the source of this variability that leads us to go in one direction or the other one? With this idea that can we increase the robustness of the hearts? Can we increase the resilience? And if damage eventually occurs, can we rejuvenate? And then lastly, and then along the same concept, neurosciences, this idea of resiliency and repair. I think we now understand that the brain is actually very malleable and able to repair itself or repair circuits as they get disturbed. And so how is it that we can understand that process of resiliency? How can we identify biomarkers that would tell us that something is going to go wrong later down the line, so that we can have interventions in place and increase that window in which you have resiliency, so that you don't have to spend a lot of time in the repair and the disease side of the spectrum. You can imagine if you can have a biomarker and interventions that allow the onset of Alzheimer's to happen several years after where it's supposed to occur. It's just the advantages of that are just from the economics of it and the well-being of individuals is major. All right, so with the last set of minutes, I just kind of wanted to cover a couple examples of people who are doing some pretty interesting and amazing work in the area, starting from basic scientists that are beginning to move into the translational arena. And then more and more, we want to try to encourage those individuals to do this by finding the right partners. And so I just want to start with a short story with Simon Gregory. He is a faculty member in Program of Genetics and Genomics. He's a geneticist by training. He's now also in the Department of Neurology in the Duke Molecular Physiology Institute. His research portfolio is pretty vast, from cardiovascular disease to multiple sclerosis, and also into autism and other diseases of neurobehavior. And so we all know about autism, and this is a restricted repetitive patterns of behavior. This is an area that we're all quite aware of. 1 in 70 children are affected by autism spectrum disorders. There's really no approved treatments to address the core social deficits. For any treatment that is available, there's a range of side effects. And there's conflict between prescription age and restrictions. And there's really a lot of opportunities for novel treatments if we understand a lot more about the disease, how it works, and also understanding biomarkers, so we can have a way of predicting whether our interventions are working or not. And so some of the work that Simon has been focusing on centers on this receptor for oxytocin. There's a lot of data out there where some kids respond well to oxytocin, some do not, and people don't quite understand it. But a lot of genome-wide association studies have been done, have pointed out to oxytocin-related activities, and the oxytocin receptor as being risk alleles for developing autism. So that means that to some extent, and this is going to be known for a while, that there's a genetic component as to whether a kid develops autism or not. But it is not as simple as that, because it is not really a type of a Mendelian trait that you inherit very easily. And so what Simon and his colleagues have identified over period of time is that there's actually a regulation of gene expression that is independent of the DNA sequence that may be participating in this process. So this is a process called epigenetics, where you can have modifications of DNA or histones that can turn promoter areas of genes into either a closed or an open conformation. So that means that really what is present in your DNA by itself is not necessary to determine an outcome, but it means the environment and how it modifies these epigenetic modifications in DNA that may determine whether you are more or less sensitive to disease. And so one of the things that Simon and his group identified is areas in the promoter region of this oxytocin receptor that are subject to modulation by epigenetic markers, both cytosine methylation and also acetylation of histones. So there's markers that they've been identifying through time, and they correlate with the expression of the receptor. And they've been looking at patients with autism and looking at markers of methylation of the oxytocin receptor, both in peripheral blood but also in the temporal cortex. And then they found that there's really this very nice, strong correlation between elevated methylation and decreased gene expression of this receptor. And so he's partnered with Dr. Lynn Sinkage in the Autism Center to begin to run clinical trials to try to figure out, what happens when you begin to intervene, what happens with patients who are in oxytocin treatment, and really develop more sensitive biomarkers, molecular biomarkers of the link between epigenetic modifications of this particular receptor. The second story has to deal with a young faculty member, Chris Wood, in the program of pharmacology and cancer biology. And Chris is very interested in signaling pathways related to growth regulation. And we all know that these are very tightly linked to cancers. Most cancers just have an uncontrolled growth, mostly because of activation of various signaling pathways, whether things like the RAS MAP kinase pathway, ERK pathways, Notch pathways, but also downregulation of pathways that usually lead to the death of cells that are hyperproliferation, like the apoptosis pathways and PI3 kinase and mTOR pathways. So one of the questions that Chris has been asking is this idea of drug resistance in cancers. Why is it that some cancers become drug resistant? And so he created a very neat little set of tools to study in a global manner how cancers respond to drugs. So he created a panel of cell lines where he had activated every single known pathway related to cancer and test them against drugs. So one thing that has been known for a while about colorectal cancers and other cancers that are sensitive to inhibitors of the epidermal growth factor receptor is that they become resistant very quickly to the drug. And so he looked at a collection of cell lines to try to figure out, what kind of pathways that get activated may compensate for this activation of this inhibition of the growth factor receptor. And I hope you can see over here is that a particular set of cell lines became highly resistant to the inhibitor, and this all had activated RAS and ERK signaling pathways. So then what he reasoned is that, well, what happens if we inhibit both at the same time? Could we prevent the emergence of drug-resistant cancers? And that is essentially what he found. So if he has a tumor that is mediated by the EGF receptor, and he treats it with a drug that inhibits the EGF receptor, it blocks cancer growth, but after 100 days, you begin to see emergence of drug-resistant versions. Now, through his analysis of cells, he figured out that if he were to inhibit the ERK signaling pathway, he should be able to inhibit the rise of this drug-resistance clones. And so it turns out that if he treats with both of them, he never gets a drug-resistant clone popping up. And this is also reflected in animals. So if he injected mice with these tumors, it is a kind of more relevant system, he finds that if he does the combination, that you never get resistant tumors popping up, but he would if he just using one combination versus the other. So this obviously has potential therapeutic implications, where perhaps when you treat certain types of tumors, you should treat right away with both classes of drugs, as opposed to trying to wait for one to fail before you introduce the next one. And then very quickly, the last example has to do with very complicated classes of tumors that have activated PI3 kinase pathways. More often than not, we can identify what the event is that led to tumorigenesis, but through time, a lot of these tumors accumulate other mutations that lead to the activation of pathways. So there's some of these tumors like this PI3 kinase that if you were to inhibit with a drug, you still cannot prevent the tumor from growing. And the assumption has been that there's probably another pathway that gets activated at the same time. And if you were to inhibit both with drugs simultaneously, you should also prevent the growth of tumors. And so this is kind of what he found is for PI3 kinase dependent tumors, that if he treats with an inhibitor of an anti-apoptotic protein, that he can have a synergistic effect. And in fact, you can find if he treats with both at the same time, he can completely obliterate the ability of tumors to form. Okay. So those are just two examples. There are really dozens that I could cover. Many of them started as basic science questions, and it's not only until they found that there was a path to the clinical world that they reached out. More and more, I would like to see some of this information flowing back, right, where the clinical problem gets presented, and then the basic scientists can come in and say, this is an interesting question that we should try to tackle. So I'm going to stop there, and I'll be happy to take any questions. Don't ask me too much about the science because this is not my stuff.
I have a question. So thanks, fantastic talk, fantastic overview. So good news to know there's receptiveness to basic science questions that might originate from a clinical environment. What are your recommendations for clinical investigators who do have a question that comes up from perhaps a clinical encounter scenario and are interested in developing a research proposal that might benefit from some of the basic science expertise that we have here at Duke, or the resources that we have, the new proteomics, genomics resources that you mentioned? What's the best process for efficiently identifying those resources and making those connections to move the proposal forward?
Yeah, so that's a great question because we don't have really an established framework to do that. Right now, it is very much word of mouth, right? It's like who you know. I know somebody who may know somebody who may help you. I would say that I am one of those resources. So in as much of a question comes up and you think, I think somebody should look into this, I would be the kind of person who will put you in touch. Mark Stacy, as the Vice Dean for Clinical Research, he can also point you to individuals or ask me, and I can find the right people to connect with. The division chiefs, the department chairs are also a good first starting point, but eventually they probably will end up routing back to me. I mean, one of the challenges is that sometimes you only know what you know, right? And so by putting you in contact with more and more people, then you get more information coming in. And there's some, we tried to get this program started called Enable, and this is by a couple of chairs of the clinical departments where we could have a formal way of having clinical faculty embedded in basic science department, kind of like a mini sabbatical. And that was going to be funded by the PDC. They started a couple of them, and I think there is going to be impetus to maybe get that restarted. That would be another way that somebody has like a great set of clinical samples and is just finding a home where they can sit down, immerse themselves, and then learn. Mm-hmm. Yeah.
Very fascinating talk. I have a quick question. So I guess in the clinical world, there's more recognition that replication and validation of existing results are becoming more and more important, as evidenced by open data sources and other centers using data and like replicating and validating findings. And you talk a lot about innovation, so the generation of new knowledge, but there's probably like a lot of knowledge that we don't know that may have been coming in from one or two labs that are driving a lot of things, but we don't know, can this be replicated or validated? Is there any emphasis on that in the basic science world to have a focus on this very important aspect?
Yeah, so there's a broader question of reproducibility of data that is at the forefront of everybody. It is studies that have failed to replicate tend to they tend to be pruned out with time. I mean, so the way the basic science culture has always been is that, you know, you get a finding, you build upon that finding. Other people outside are going to look at the finding and they're going to try to build upon it. And if they can't, then there's this back and forth of why is it I couldn't reproduce what you saw? And more often than not, you will find that it is not because any one lab did something wrong, but there is, they're coming in from a different angle. And at the end, there may be backs and forth, and then until you get a consensus that emerges. The problem happens, I think, when you suddenly end up going into a preclinical route very quickly without having a lot of people doing this back and forth. So there's a disadvantage. The disadvantage of not having this back and forth is that you don't get to the patient as fast as you can. And the other disadvantage of not having this back and forth is that you not devote a lot of resources going down a clinical path before you wasted a lot of time and resources. So, you know, the other thing I should warn people about, you know, because there's been a lot in the news about, you know, some company did a study, is like, well, you know, X% of studies out there are not reproducible. If you reanalyze the data, you find out that data was not reproducible either. So depending on the statistical analysis that you do. A lot of times, there are enough nuances in experiments such that it's not very easy to reproduce any one result. I know this is not reassuring news, and I wish I had a better answer to your question, except to say that it is, science is a little bit messy. The more you can do to build in controls and repetitions to make it as robust as possible, the better off you will be. The more we understand all the parameters that change what we get out of an experiment, I think the more reproducible we can get. You know, there's plenty of examples where things work extremely well in cell culture, will work very well in preclinical models of infection, and then you go into humans and it all falls apart. Because there is not flaws, but there's limitations to how you do your experiments in any one system. And the complexity of going into human is, I think we will need better framework to maybe checklists of what we need to do to move from a preclinical to a clinical setting. I know it is not a very reassuring answer, but that's what I have.
Thank you very much.
Sure. I finished with a tough question.